Hello and welcome to our Friday Flip Day for December the 2nd. Some of you may be watching this on Thursday the 1st, and if so, happy Thursday. I do want you to think a little bit about what's due tomorrow, homework 9. We'd like to get that finished off for tomorrow, and I will certainly be around during Open Lab if you'd like to work through some of that. Homework 10 is a single question where I'm simply asking you to relate the topic from which you learned the most and the favorite topic, and of course I do want a substantive response to that question, but by no means do you need to write me a book. I am hoping to finish up the final exam on Saturday. I'll post it on the evening, Saturday evening, and then you can get jazzed and get sexy-minded with it. I hope you will really enjoy working through it, and of course we will already have been working through it because this Friday we have our flip day, and we worked through a question earlier on our last Friday flip day, so we're already taking that on and enjoying it even as we speak now. So we need to complete the abstract packet early next week, so please do submit me that revised version if you need to make final revisions. Many of you have already heard back from me and that you have rocked it and you are done, so know that you are already ready to go and that you don't need to worry about anything for the abstract packet. But most of you haven't sent me a full version of your poster yet, so do think about trying to do that by Saturday night. That would be wonderfully convenient to you because that would enable you to focus your time on your final exam. I did want to make a quick note that the STEP Tutoring Center is hosting a finals extravaganza. So if for some reason you have gotten to love a tutor over in the STEP Center and you want to get extra help from that person, do bear in mind that they will be extending their hours for December the 11th, Sunday 1 to 9 p.m. And you can check their website for other resources that they're offering in preparation for final exams. Of course, things are a little different for us because you'll have your final exam, but that doesn't mean you don't still want to seek help from the STEP Tutoring Center. I'd like to jump in and in beginning our coverage today, I know it sounds sort of funny, but I want to take you to a Turkish delight store. If any of you have ever had the chance to go to Turkey, wow, what an amazing place. And I can just imagine bringing all 120 of you, all of 125 of you and saying, look at this Turkish delight, let's check it out. And here's your assignment. I want you to come up with a way to categorize or classify the different types of Turkish delight. So I can see you ravaging on in and deciding, some of you, to classify it based upon color. Maybe you'll find all of the ones that have kind of a red tinge to them, and you classify all of those together. And then maybe some of you would think, no, I am totally going to classify them based upon shape, you know, and others are going to say, no, size is the way to go, and you put them in order of size. Maybe some of you would get really jazzed and you would say taste. Taste is the only way to go because, you know, you can't judge it by its package. You want to know what's inside. Or maybe consistency. And then I have a feeling if I told you to argue about whose classification scheme was the best, you'd be able to do that as well. So you would say, you know, my classification scheme trumps your classification scheme because it looks at not just the external characteristics, but it also gets to the internal characteristics. So you may think, what does this have to do with microbiology? It has everything to do with one particular discipline within microbiology called taxonomy. Taxonomy is the science of grouping things, putting them into groups in order to study them, in order to better understand them. We think that way, don't we? We like to group things for better or for worse. Sometimes we put things into groups. Now in science, this is called taxa. And what we just did, the exercise with the Turkish delight, is not very much unlike what scientists have been doing for many years. And we've experienced it in the lab. Remember that we started by asking you to think about classifying bacteria based upon their shape. So we talked about bacilli and cocci. And then we said, well, we could also classify them based upon their cell wall structure. So we started to think about things like gram-positive cells versus gram-negative cells, and then talked about acid-fast cells and flagellated cells, and how we could use those features to distinguish or group these organisms. And then later we began to talk about other ways to get deeper into the kinds of groups that we might be looking at, and we could think about whether or not something has a certain sequence of genetic material. So we could think about the genotype, the genome of a cell, and we could connect or compare and contrast the different types of DNA sequences that we see across the prokaryotic world. So you can see why scientists might also argue that their grouping was more suited or better. We've talked about using ways of generating energy, whether or not something is a chemo-organo-heterotroph, whether or not it utilizes certain energy sources as their sole carbon source. And we've grouped it based on biochemical characteristics. So there are so many ways in which the analogous grouping of Turkish delight is paralleled by what we do in scientific groupings within the particularly within the bacterial world, and that's where we're going to focus our attention today. So when we think about taxonomy as a study or a discipline can be separated out into three different categories. Of course, identification, which is what we've been focused on in the lab. You've been working on how to ID bacteria, and you've done that by using phonetic characteristics. That is, you've looked at the phenotype of bacteria, and you've been able to use that as a means to identify them. And then once we know their identification, we can work on how to put them into groups or taxa. That's where classification comes in. Classification means grouping organisms based on some sort of character relatedness. Maybe it is based upon the kinds of energy sources that they use. Are they photoautotrophic, chemo-organo-heterotrophic? Some sort of way of grouping them together based on some character or similarity. Lastly, the final area of taxonomy is nomenclature. Now, nomenclature is not something we're going to spend very much time on. There is a large document of rules on standing nomenclature. It says it's got to have the particular Latin prefixes and suffixes. And there's some rules, like it used to be when Theodore Escherich was around, he could name bacteria after himself. That is no longer something that you can do. So we'll use a hypothetical, fictional example to help us better understand this. Say that maybe Greg and Connor go on an extreme caving expedition. You two would totes love that. And you find some new bacterium. You believe that it's a bacterium rather than an archaean, but you don't know for sure what it is. You go through this process of identification, and you find out that you don't find any hits at all in GenBank, which means that you believe that you have discovered a novel organism. You're now ready, after going through this process, you're ready to name your organism. You're not going to name it after yourselves, but you may name it after the cave in which you discovered it. And then that would allow you to link it, right, to perhaps that origin, that area of origin. And that's often done with the species name. We're not going to belabor this process of nomenclature. Know that you can find the rules for standing nomenclature. You can even find those online. So we're going to turn our attention to looking more deeply at the two categories of identification and classification, knowing that we're already pretty well versed in one of those, identification. We've used the phonetic or phenotypic characteristics of bacteria in order to put them into groups. We've been doing that for a while now in lab. In fact, we turned away from it, but you've still been working to write your report. Many of you have just turned that in. And in that process of using phenotypic characteristics, you began by doing a lot of microscopic investigation. You did some gram staining. You were able to differentiate organisms into two different groups that way. You were also able to do a lot of biochemical testing, and that really was the nuanced piece of grouping that allowed you to get down to the identity. But of course, that was coupled with the cultural growth characteristics. And I know many of you even use something like odor to help you identify your unknown. Those of you who had the epically phenomenal Pseudomonas aeruginosa, you will have smelled that, and you have known that it smelled like artificial grape flavoring. And you would have also observed its growth in broth, and you would have recognized that it formed that beautiful pellicle or surface membrane in the broth, and that's also indicative of Pseudomonas growth characteristics. Additionally, when it was growing on the plate, you will have noticed that the colonies were highly mucoid. In fact, at times they may have been difficult to bring off of the plate to get into solution in your culture. So these were phenotypic characteristics that all came together to enable you to get an identification. However, as it turns out, on Greg and Connor's extreme cating expedition, it's unlikely that the novel bacterium that they discovered was a bacterium that could grow in our growth media in the lab. In fact, we can culture and characterize less than 1% of the bacteria that are out there. And so we actually have a very, very small view of the microbial world based upon our limited capacity to culture and grow organisms. So that's where we have had to turn much more to genotypic characteristics in order to identify organisms. And this is a much more powerful technique using nucleic acid analysis. So with this sort of technique, we need to take some time to look at the nuanced techniques that exist for us to assess sequences of nucleic acids and get at these identities of organisms that cannot be cultured and characterized in the lab. Now, some of you may have looked at this list and said, you know, the truth is that when I go to the doctor and the doctor says, oh, it looks like you have strep throat, the doctor rarely, if ever, uses one of these two techniques. And in fact, many times the patient's symptom is what the doctor is using to diagnose or to, quote unquote, identify these bacteria. Now that has some major problems because if an antibiotic is then prescribed, we actually haven't done a Kirby-Bauer assay to determine if that organism is what the doctor thinks that organism is. So on the final exam, I tell a story about being in the hospital with one of my athletes. We had to take her to the ER during Thanksgiving break. And they cultured with the thought that she might have strep throat. They did the rapid strep test, didn't see a positive, and so then they cultured it out onto a plate, a blood agar plate, and they were going to go through the long, identification that we did in lab. That was some very good healthcare practice because they were confirming that what they had seen as a negative on one test was actually negative, and they were actually culturing it up. But the problem is that many times money is saved by not doing that, and so antibiotics are prescribed based only on patient's symptoms and not on either of these other identification means to determine what the culprit of infection might be. So let's talk about why classification within the bacterial world is super complicated. Now, those of you like Emily who are really knowledgeable about larger organisms, that is maybe about horses or cows, you know that when we talk about classifying an organism into a species level, we are typically looking at the ability of two horses to interbreed with one another and then form fertile offspring. That is how we define species. So we can have that means, that simple means. But because in the bacterial world we have a lot of complication with that, not only do we have similarities in size and shape, but we also have that inability to reproduce sexually. And so because of that, we can't define species the way that we define species for organisms like horses and donkeys, right? So because of these difficulties, we tend to rely upon genotypic similarities. That is, we tend to look a lot at phylogeny. Phylogeny is the sexy-minded term for evolutionary relatedness. So this is an extremely useful thing in the prokaryotic world. So when it comes down to classification, we tend to utilize that a great deal, and we're going to talk more about that shortly. But I want to make sure that we're all understanding that there's a lot of lingo that comes up when we start to talk about taxonomic classifications. As is true in the world in general, where there's groupings, there's often hierarchies. And this is true within bacterial taxonomy as with any other. So you all have become very familiar with the lower rungs of the taxonomic hierarchies at this time. That is, we've been talking for a long time about genus and species. Now, we've also talked a bit about strain designation. We know that the strains that we work with in the lab are those that are generally non-pathogenic laboratory strains, and we've talked about the difference between E. coli K12 that we work with in the lab versus E. coli 0157H7, which we know to be a pathogen. So these are the lower rungs of the taxonomic hierarchy. A strain comes below species, and so we could define species as a group of related isolates or strains. It's interesting to take a moment to note that we could actually dissect strains down even further into biovars or morphovars, and this is actually something that could be even more homogeneous. So that's one way to think about hierarchies, is that at the lowest rung, these hierarchies are the most homogeneous. That is, they're most similar to one another. Now we could look at the species and use an example such as Escherichia coli. Remember that coli is the species designation. So we remember that originated from the fact that it is isolated in the colon. Now, if we go to the next level up, we all know that that's genus. So it's pretty easy to start to define these hierarchies because they're just a collection of the related group below them. So a genus is a collection of related species. And of course, we know Escherichia is the genus name. Now, you might think to yourselves, well, really, why is this necessary? Because, I've never heard of any other kind of Escherichia than Escherichia coli. That's probably true. You maybe never have heard of them, but they do exist. There's Escherichia fergusoni and Escherichia alberti. So there's more than one species of Escherichia. Now, the next highest level that encompasses a group of related genera is family. And you know this well too, because in lab we talk all the time about the Enterobacteriaceae. The Enterobacteriaceae, that is the family that encompasses E. coli, but it is also the family that encompasses many of your other gram-negative unknowns. For example, some of you had Shigella dysenteriae. It's also in the family Enterobacteriaceae. So is Proteus mirabilis. And in fact, most of the gram-negative unknowns, except for Pseudomonas, fit into this family. And we'll look at that on a visual here in just a moment. Now, what's really cool is that family is always easily recognizable in nomenclature because it always has the suffix of ACA. So if you look for that ACA at the end, you can say, oh, that's a family. And so you might guess that Pseudomonas belongs to a family called Pseudomonadeaceae. So there are different families, but we can always recognize them based upon the ACA at the end. So Enterobacteriaceae encompasses a great number of our gram-negative unknowns, ranging from Enterobacter to E. coli to Salmonella to Shigella, and all of the ones that we had worked with in lab except for Pseudomonas. But remember that this isn't the most heterogeneous group yet. That is, we can keep on going up in these taxonomic hierarchies to order. Order is recognizable as well because it always ends in alis, and maybe it's not too much of a stretch to realize that Enterobacterialis is going to be the name of the order that encompasses the family Enterobacteriaceae. So there's more than one family in the order Enterobacterialis, and likewise there's more than one order in the class that's going to be the next rung up, a collection of similar orders. And this always ends in IA, and in this case these are the gamma Proteobacteria. So the gamma Proteobacteria now encompass all of our gram-negative unknowns. So this is crazy because gamma Proteobacteria, that's only one class within the next highest group, which is a phylum or a division called Proteobacteria. So this is crazy because we have now encompassed a great number of gram-negatives that we have learned about in a very tiny itsy-bitsy piece of what is just one phylum within a much more biodiverse world of bacteria. Now everyone knows what's at the top, right? Because we've talked already about the three-domain system being comprised of eukarya, archaea, and bacteria. And of course here we're talking about bacteria. So the domain in this case is bacteria. But I could sort of silently hear somebody shouting out, maybe Nathan. He was like, kingdom, kingdom. And in fact, many of you probably learned the 5 kingdom system for life, but really it's antiquated. And in a moment, we're going to talk about the contribution to changing that was made by a man named Carl Woese when he proposed this 3-domain system in the 1970s. So kingdom, it's not missing, it's just kind of in flux right now. So all of this is well and good, and sometimes we are good about talking about bacteria in their taxonomic hierarchies and talking about gamma proteobacteria. But more often than not, we tend to use a little bit of informal grouping, and you'll notice on the final exam, I talk a lot about the lactic acid bacteria. Well, there are lactic acid bacteria in multiple different classes, but that being said, we put them all together because of their metabolism, right? They all make lactic acid. They all ferment the milk sugar lactose, make lactic acid. So they go together in what we would consider an informal grouping. We could also think of the sulfate reducers. And just the other day when Parker was dissecting his Winogradsky column, he noticed a pocket of black. Well, remember that those are the sulfate reducers. They produce hydrogen sulfide gas when they use sulfur or sulfate as a terminal electron acceptor in anaerobic respiration. So sulfate reducers, another group. We could also talk, of course, about the chemoorganoheterotrophs as a group. We could talk about many different informal classifications that aren't necessarily paralleling the more formal hierarchies we see here. So you could say that there could be 2 things that are sulfate reducers, but they're not even in the same order. And that's commonly the case. So this is a tough one because I think we like a lot to talk about things that have metabolic similarities as a group, and yet phylogenetically, based upon their genotypic sequences, they may not be closely related. This creates a bit of a crux, I guess. That being said, I want to take a moment to talk a little more phylogeny. So when we talked about the fact that one of our gram-negative unknowns, so for example, some of you had Shigella dysenteriae as your unknown. It is just one of many different species of Shigella that then belong in the genus Shigella, and the many different genera ranging from Enterobacter, which some of you had, to Klebsiella and Proteus, which we talked about, being all different genera that all belong to the Enterobacteriaceae family. And then the Enterobacteriaceae family is one family in the order Enterobacterialis. And then we start to see all of these other orders. But hey, look, here's where we catch Pseudomonadalis, which was in the family Pseudomonidaceae. Now, so up until this point, we were in different branches for our Pseudomonas unknown versus all of our others. But now we've captured it up in the fact that they both Enterobacterialis, the order, and Pseudomonadalis are together in the class Gamma Proteobacteria. Now we can see, whoa, check it out. All of our gram-negative unknowns are Gamma Proteobacteria. And yet there's beta proteobacteria, alpha proteobacteria, delta and epsilon proteobacteria. So there's this whole world of other organisms that we haven't even touched in our unknown identifications. And then, of course, these are all members of the phylum Proteobacteria and the domain Bacteria. And we haven't even considered the whole domain Archaea. So the world of the microbial world is vast, is it not? Now, maybe you're wondering, how in the world am I going to remember all of these taxonomic hierarchies? Well, there have been some great mnemonics that have been developed by your fellow students, and I'm going to share some of these because they're absolutely awesome. So they help you just to recall them. Dave put his class in order for grading simplicity. That helps you remember domain, phylum, and this is kind of cool because class and order are the same. Family, genus, species. Dutchmen physically clash over famous German skiers. That's another one, good one. How about this? Dirty people collect OxyClean for getting smelly. And my absolute all-time favorite, Don or Donna, please come over for great sex. You will never forget the taxonomic hierarchies again. But this allows us to talk about a man who I believe may be one of the most sexy-minded individuals of our time. Carl Woese is a molecular biologist turned physicist, and up until just a couple of years ago when he died, he was working feverishly on classification of the microbial world and really of our entire world. So in 1970, Carl Woese proposed that we utilize an ethically special molecule to enable us to design a new classification scheme. That ethically special molecule was ribosomal RNA. Now, let's think about why ribosomal RNA is so special. Well, for one thing, ribosomes, they're essential. Ribosomes have to be able to function, or else the cell cannot make proteins. This is epically important. So one would note, accurately so, that ribosomal RNAs must have certain conserved sequences that across all life have to stay conserved in order to maintain functionality of this molecule. And if those are lost, then of course the organism dies. If there are mutations that compromise that central function, then this is lethal, right? Those would be lethal mutations. So there are maintained sequences across ribosomal RNA, but there are also very special differentiated sequences. We think of them as fingerprint sequences that very much differ across organisms. That makes our ribosomal RNA very special because we can track both similarities and differences across time and across organisms. And so the particular ribosomal RNA that was chosen was 16S ribosomal RNA. And all of you will remember that the 16S ribosomal RNA is the one that catches the Shine-Dalgarno sequence, right? It recognizes it. So 16S ribosomal RNAs are what were chosen to sequence and then to create a 3-domain classification system for life. Now, this particular picture, it was one that I took from Carl Woese's webpage when he was faculty at the University of Illinois Urbana-Champaign. And this shows, I think, very succinctly the 3-domain classification system, where you can see bacteria, archaea, and eukarya. Now, we know that the Earth is thought to be some 4.6 billion years old. We know that some 3.5 billion years ago, there was a fossil record. We recognize that that was, of course, the Archean chert that we've talked about in the past, but that from there, about 1.4 billion years ago, eukarya diverged from archaea. And this is where things get super interesting when we do focus on this particular 3-domain classification system, because you'll notice that archaea, which branch here, bacteria, well, archaea are related to bacteria, but look, they're also very much related to eukaryotes. And this is where we start to very much see the argument that I brought up at the very beginning of class as to, should we have the term prokaryote? Really? When we've come to Carl Woese's classification scheme, and there are many others, but this is the favored one as the favored scheme for classification. Does it really evoke the need for the term prokaryote? Because really it doesn't. Because here we've got archaea that are very distinct from eukarya, which are very distinct from bacteria. Is there cause to group together archaea and bacteria, except for to say, well, they're not one of us? Probably not, which is why you hear me try to say bacteria, archaea, and eukarya rather than always talking about prokaryotes. But that's a term that's going to die hard. Who knows if it ever will, in fact. So one of the critiques that Carl Woese took about his 3-domain classification scheme is that it didn't account well for lateral gene transfer. This is true. It doesn't account well for that. Really, it only looks at ribosomal RNA, and that's the only thing that it tracks. So there have been some that have countered his scheme. And I should say also that Carl Woese, in the last of his career, thought all the time about lateral gene transfer and about how to, in fact, improve classification with that in mind. But this is another phylogenetic tree that I want to bring your attention to. And this is one that I think, oh my goodness, it is so beautiful. This was just published last April, and it really gives us this idea that we are coming to understand that life in the bacterial world is far, far, far more eclectic and diverse than we ever knew. So check it out. This whole purple region right here, up until very recently, was not even recognized as candidate phyloradiation. This all is due to our ability to better utilize genotypic data to understand what is present. So our ability to sequence, to utilize 16S ribosomal RNA and sequence much more of what's out there, say in our extreme caving expedition, to really get a handle on the vastness of what is there. We can't culture any of this, and so its existence was only made, knowledge of its existence was only made possible by these genotypic techniques. So this is a really fantastic figure, one that I say go find it online and recognize that this was published in Nature Microbiology on April 11th, and recognize its incredible, and in fact, even now, today, this would be changed a great deal because new species are being recognized every single day. So new forms of life in the bacterial world, that's really cool. This is another phylogenetic tree that kind of argues here's how we should be considering the complexity of horizontal gene transfer, lateral gene transfer. And I would even say that just this notion of plastids and mitochondria being the only link between archaea bacteria and eukarya may be oversimplified, because there's probably some arguments that that more types of lateral gene transfer are possible. This one also further complexifies this situation by looking at more than one primordial ancestor, and that is something that is not done in Carl Woe's phylogenetic tree. So a multiplistic or polyphasic technique for forming phylogenetic tree might be a more accurate technique. That is, maybe we look at 16S ribosomal RNAs for certain things, but then as we see vast similarities within a particular group, maybe we then diversify to look at other housekeeping genes as further differentiating organisms. So a lot of complexities to think about there. So we've now understood that identification, classification, and nomenclature are the central pieces of taxonomy. And when we begin next time, we will be looking at different specific techniques for identification and classification. And we'll be kicking off by looking at a way in which the kinds of phylogenetic, a way in which the kinds of phonetic or phenotypic ID that we've done with biochemical testing can be used as a classification scheme as well. It takes some time and energy and some vast computer databases, but it is a possibility.